Okay, so my name is Isobel Araujo.
My name is Gentry.
And our project is called the Global Greenhouse. So some background information before we start. Our project has a lot to do with the atmosphere and changes to our atmosphere. And so different layers of our atmosphere protect the Earth from solar flares, asteroids, UV radiation, gamma rays, and other harmful wavelengths of the electromagnetic spectrum. So as well as those layers, other layers of our atmosphere keep the Earth insulated and habitable. Infrared light warms the Earth to allow life to continue on our planet, and the greenhouse effect occurs when light from our sun penetrates the atmosphere and radiates off the Earth's surface, and the heat gets trapped by the atmosphere and warms the Earth. This is a picture of the greenhouse effect, and as you can see, the sun shines down on the Earth, and some of the radiation is reflected off the clouds, and some is absorbed by the Earth. And and sometimes it escapes through the atmosphere, but when it's radiated, the radiation is absorbed by greenhouse gases that causes heat. And so our things like deforestation, cars, and factories all contribute to the greenhouse effect to create sort of a more man-made greenhouse effect, which is not exactly a positive thing. Global warming. Humans are changing our planet, and not in a good way. The Earth is heating up due to recent rises in CO2 emissions from humans through cars, factories, things like that. It's not a natural greenhouse effect. It's a man-made greenhouse effect, which is bad. It's causing our atmosphere to be too hot. The average global temperature has gone up by 1.4 degrees Fahrenheit since 1880, and it might not seem like a lot, but it makes a difference. And this is the fastest increase in global temperature in the past 600,000 years. And you might wonder why is this relevant? Why do we care? Well, global warming can cause extreme weather at the risk of the lives of many plants, people, and animals, and also their habitats. Global warming can lead to species extinctions and habitat shifts for native plants and animals. It can cause severe drought and floods in important areas where we grow our food and cause famine, and that's bad. Global warming is getting worse. So our experiment used different greenhouses that we made to simulate possible future atmospheric conditions resulting from global warming. So we wanted to explore the effect of extreme weather and extreme climate on various crops as well as native plants. Our hypothesis was that if different greenhouse coverings were used on plants, the thickest covering with no holes will have the most negative effect on the plants within that greenhouse because it would represent the thickest and toughest atmosphere for plants to live in. Our second question was, how would the health and development of crops compare to native plants when both are inside the greenhouse? And we hypothesized that the crops would thrive in the greenhouse while the native plants would not, because native plants in this area are used to dry, warm climates and not the hot and humid tropical climate we anticipated inside our greenhouses.
Experimental design. As a class, we built 4 different greenhouses, each with varying thicknesses of polyethylene tarp. We used 6 mil solid, 2 mil solid, 6 mil with holes, and 2 mils with holes. By the way, mils means 1/1000 of an inch. We also had a negative controlled greenhouse, which had no covering. This represented the present day climate. This is a picture of my partner Isobel standing by our bed of greenhouses. This is the 2 mil with holes. This is the 2 mil solid. This is our control group, 6 mil with holes and 6 mil solid. We used crops and native plants. The crops we used were corn, tomato, and radish, and the native plants we used were black-eyed Susans, Indian grass, and Japanese silvergrass. Our procedure is we dug up one of the beds in the children's growing garden. Next, we measured out plots for each greenhouse. Then we came up with a design for plant organization to go in each greenhouse. And finally, we assembled the greenhouses. Each day at noon, Isobel and I would go out and check the CO2 levels, temperature, and O2 levels. And at the conclusion of our experiment, we weighed each species in each greenhouse and took the average biomass of each plant species for comparison. Our materials are, we had different size tarp. We had a lot of plants, actually. That's the list. We use a lot of duct tape, PVC, a lot of duct tape. We use a lot of different size PVC tubes, and T-joints and elbow joints for each bed. And we had a CO2 monitor, a temperature monitor, and an O2 monitor. For our temperatures at noon, we found that the greenhouse with the 2 mil solid tarp was the hottest because the light was able to penetrate the material, but infrared light, which is heat, was trapped in there. For our CO2 levels, we found that both solid coverings had the highest CO2 levels due to the fact that CO2 was trapped in the atmosphere of the greenhouse. And for our O2 levels, we found that 6 mil solid had the highest oxygen because the gas was trapped within the hoop house with nowhere to escape. The more CO2 and O2 being sucked in by the plants is an indicator of photosynthesis, which means the plants are growing more cells.
So at the conclusion of our experiment, we harvested all the crops that we had grown from seed. And so this is the graph for the radish biomasses. We took the above ground biomass, which is the leaves, and the below ground biomass, which is like the tuber part. And we found that within the greenhouses, the 6 mil with holes greenhouse had the radishes with the highest biomass of both the above ground and below ground biomasses. And you can see our corn also showed the same result. It's very obvious to see that the 6 mil with holes environment was the best for the corn. This suggests that corn likes hot and humid environments, and this might be because corn can do a different version of photosynthesis, which helps it in this climate. Because we couldn't take the biomasses of the native plants because we did not plant them from seed, we ranked them from healthiest to unhealthiest by observing them and seeing if they had brown leaves or if they had flowered or not. And you can see that all the plants with the highest ranking all have holes in them, or all the greenhouses had holes in them. So that's interesting.
Pictures and qualitative measurements. This is pictures of the plants inside of our 6 mil solid. As you can see, the black-eyed Susans are dead. And right here, we had an error with our water hose. It flooded our corn, so that wasn't too great. These are pictures of the 6 mil holes. Again, I think this is the Indian grass. It's slowly dying. It has brown everywhere. And the other plants are doing well. This is a picture of our control. The Japanese silvergrass isn't doing too well. The black-eyed Susans kind of in between, and I think this is radish. Yeah, this is doing okay. For our 2 mil solid, the black-eyed Susans aren't doing well, nor is the Indian grass. 2 mil holes, it's obvious that the black-eyed Susans are dead, and the radish and the corn are doing okay.
Okay, so in conclusion, our greenhouses modeled the advanced stages of global warming by creating environments with higher temperatures, higher humidity, and elevated levels of CO2. Especially the solid greenhouses, they trapped a lot more gas, and everything got hotter and had higher CO2 levels. Just a side note, normally in an enclosed environment with plants inside, we would expect CO2 levels to drop and O2 levels to rise as the plants are sucking in carbon dioxide and giving off oxygen, but our results showed the opposite. This could have been because of insects, bacteria, or other organisms that got into our greenhouses and were giving off CO2. Our first hypothesis was not supported. The 6 mil solid covering was not actually the worst environment for our plants. The 2 mil solid covering turned out to be the worst because it was a thinner material, so the light could come through, but it didn't have any holes to allow gases to escape, so everything inside that greenhouse got really hot and everything died. Our second hypothesis was supported. Through our observations, the native plants did not do well in that climate at all, while our crops were able to thrive. The native plants need an environment where, in this area, the native plants need an environment where it's dry and warm, not tropical, like it was inside the greenhouses. Our experiment indicated that global warming could lead to the elimination of many of our native species and the ecosystems that these species live in. And if this happens, much of Earth's natural resources could disappear. Some problems with our experiment are things that we would like to perfect in the future. We had some problems with our watering hose. There was a big hole in it. We had problems with planting the tomatoes because some of them didn't grow and some of them didn't sprout, and most of them were eaten completely by bugs. insects inside the greenhouse was a problem. Bacteria and other sources of CO2 within our greenhouses threw off our data a little bit. We had some problems with our CO2 and O2 monitors, and we had to repair our greenhouses a lot.
We would like to thank our instructors, Ben and Murphy, our entire College First class, and Miss Shelley Bender. We would also like to thank our mentors, Haillee, Shamra, and Eileen, and also the donors and funders of this program.
Thank you.
Any questions? Yeah.
Oh, you. Okay. Yeah.
It looked like you used an opaque plastic. Why did you use an opaque plastic versus like a clear plastic that's more like able to level more amount of light through?
That was the plastic that was most available to us. Yeah, that's the reason why. So, you?
What was the purpose of using some of the covers had holes and some didn't have holes? What was that like, sort of as a model?
We use it to test the different effects, like, cause with the holes, air can come in. With the holes, air is trapped in there, so it won't go in or go out or come in. So we want to test the different effects on the plant growth.
Yeah, like rising CO2 levels in our atmosphere, if they're they would be higher inside the solid greenhouses. So we wanted to see what that would be like. Go ahead.
What kind of instruments do you use for CO2 and
We had
What was the question?
what kind of instruments did we use?
Oh,
They were kind of like little. They were just monitors, and we
Yeah.
plugged them into a reader, and we actually duct taped them to a ruler and stuck it in there.
yeah, we had Velcro, like, it was like a Velcro door. We could just open it and stick the monitors in.
Yeah. So we didn't want any gases to escape, so that's how we minimized that.
Took measurements.
Over there.
Can you repeat the question, please?
Which one was the best again?
The-
Or does it vary between plants?
It varied between plants. We tested each like plant and like, well, each house actually. The 2 mil holes was the worst because I'm sorry, 2 mil solid was the worst because it was thinner, so it could let in the light. Let in the light, but it held the infrared light because it couldn't go anywhere, so it was hotter in the 2 mil solid. Yes.
Mike, the more holes of the greenhouse that you have, is that going to affect the sunlight that plant get?
Oh, you mean does the sunlight come through the holes?
Yeah.
Um,
Affect the growth of plants.
Yes, but we didn't think it would change that much of the experiment.
One is that for the CO2 monitors, I think it's going to change in like several seconds. So how do you collect a accurate?
Well, we had it in the held the we held the monitors in the greenhouse for several minutes to wait till we got a stable, like at least between like 2 different CO2 levels to like get a stable level, reading for each O2, CO2, and everything. Any more questions?